Hi, today I wanted to talk to you about the research my colleagues and I have done in finding an alternative pathway to lysosomal degradation of some of the more difficult to target extracellular proteins in the human body. So let's start with a quick summary. Lysosome-targeting chimeras, or LITACs as we've decided to acronym them, are molecules that are capable of binding to both cell surface lysosomal targeting receptors, or LTRs, and extracellular proteins. In our study, we theorized that we could synthesize a molecule that would effectively induce a cell to engulf the target compound it was bound to, which would then trigger the lysosomal degradation of that target molecule. We then tested a number of clinically relevant compounds to determine if we had successfully synthesized a molecule that could actually perform such an action. And then after we had determined that we had in fact synthesized such a molecule, we tested the effectiveness of an antibody when used as the target molecule that our LITAC that we'd synthesized was bound to. The end results of our experiment indicated that LITACs are capable of successfully inducing lysosomal degradation of extracellular and membrane-associated proteins. To the left is a depiction of how LITACs work conceptually. You have the target protein bound to a lysosome-targeting chimera, which has been registered by this receptor, CI-M6PR, and then induced into the cellular lysosome for degradation. And below is the synthesis pathway of the M6PN glycopolypeptide ligands we used for the CI-M6PR receptor on the cell surface. Some of our key points and findings include that we noticed LITACs use a mechanism that is complementary to currently existing pathways for lysosomal degradation. For this study, we focused on the lysosome-targeting receptor cation-independent mannose-6-phosphate, as it's recycled through the cell and efficiently moves its cargo from the cell membrane to pre-lysosomal endosomes. We synthesized serino-mannose 6-phosphonate M6PN residues on glycoproteins to test the way that LITACs affects the K652 cell's ability to regulate the uptake of neuroadmin 647. What we noticed was there's a 5 to 6 fold increase in the uptake of the compound for cells that had been treated with the M6PN polypeptide comparative to those who hadn't, which we can see in the figure on the next slide, where the polypeptide M6PN is in red and is noticeably larger in each of these cell lines labeled across the bottom axes than the uptake of the extracellular protein I mentioned earlier in cells that only had the polyreceptor GAL-NAC. We also tested whether or not conjugating the M6PN glycopolypeptide to an antibody would effectively reprogram the antibody to direct extracellular agents to the lysosome. Our test proved that the LITAC AB1 of the study, when conjugated with the mouse antibody IgG, resulted in a 40-fold increase in the lysosomal engulfment of Alexaflor 488, which was the intended extracellular agent of that test. In addition to discovering a successful mechanism for the M6PN conjugation to an antibody, we determined that this conjugation had no detrimental effect to antibody clearance in vivo or in the cell, which is extremely convenient from the point of an immunologist, as it allows for the continual study of the cell's response to multiple different antigens and antibodies, as the cell is able to continuously clear those antibodies and maintain its usual function. This figure is the M-cherry antibody and antigen uptake in the absence of the LITAC AB1 and in the presence. As you can see, these three near negligible bars represent antibody and antigen uptake in the cell for lysosomal degradation in the absence of the LITAC AB1. There's a noticeable difference in the amount of antibody antigen compound that is taken into the cell for lysosomal degradation when we use the LITAC AB1 as a signaling molecule compared to when it's absent. So why is any of this relevant? Well, extracellular proteins play an important role in cancer, age-related diseases, and autoimmune disorders. The proteasomal conventional pathway is limited to proteins with intracellular domains and is inefficient at triggering the engulfment and degradation of certain extracellular proteins. LITACs resolve the issue of protein limitation by binding extracellular components that are difficult to recognize, so that the cell can recognize the LITAC instead of the more difficult protein. This provides an alternative pathway for the clearance of extracellular agents that have a detrimental effect on cell function. The broad application of the LITAC platform has untold implications for the manipulation of a cell's ability to degrade any number of compounds. We also found that it was possible to synthesize a number of different LITACs based off of a single type of cell surface LTR, each of which was capable of inducing the degradation of an even larger number of extracellular agents. And while we were not able to determine the exact gene sequence responsible for cell surface receptor recycling, we did determine that the mechanism is closely linked to the typical pathways of lysosomal degradation, and is thus necessary for the successful completion of the LITAC pathway for pre-lysosomal engulfment. Thank you so much for your attention, and have an amazing day.